Hello there, welcome to this section of biological molecules. It's past exam questions, and we're on this next section, which is lipids and phospholipids. So the first question, triglycerides contain three elements. M and N are formula for two macromolecules. Which of the formula M or N corresponds to a triglyceride? Explain your answer. Okay, so you're not expected to know this, you're expected to be able to work it out from what you know. So we should know the structure of a triglyceride. So we should know that it's a glycerol and then it's got three fatty acids. So the structure of the glycerol would be three carbons with the OH groups out to the side because it's an alcohol molecule, and then hydrogens on the others. And then we should know that the majority of the hydrocarbon is hydrogens and carbons. Now, we don't know how long that hydrocarbon chain is, so we don't know how many carbons there are going along here. So we can't just count them, okay? But what we do know is that when it joins, we've got a carboxylic acid group here. So we've got OH here with the double bond O and then single bond OH. We know that the water molecule comes off like that. So that part, oops, it wants to delete the whole lot. There we go. I don't think I can just delete those. So these ones come off, we'll just scribble them out. Okay, and it just joins on like that. Then this molecule is all just hydrogens. Okay, because that's hydrocarbon on the fatty acid. So the same thing you'd have here, we normally simplify it just to a zigzag line. So what we're looking for is which one of these could it be? So we've got this one, which has got 55 carbons, 98 hydrogens, and just a few oxygens. And we got this one, which has got carbons, 18 hydrogens, 30. So obviously, again, like approximately double hydrogens compared to carbons. And then we've got less oxygens than carbons on there, but not as few here. So the difference between the carbons to the oxygens here is absolutely huge ratio difference, whereas here the ratio is kind of almost the same. So we've got to think which would it be. So you could draw one out and you could count them and you could see what kind of ratio do you get between the number of oxygens to the number of carbons. Now if you imagine I've drawn all the carbons out on there and there and there, this hydrogen obviously goes in the bond. So for each one, you've got this, oops, to the carbon, oxygen up there, and then these are just hydrocarbons. So you've got this carbon, this one, this one. So you've got three there, and you've got three carbons there. Okay, so you've got 1, 2, 3, 4, 5, 6, and then you've got plus however many you've got in the chain. So you're going to have loads of carbons. Okay. Oxygens, on the other hand, you've got the three oxygens here and the three oxygens up here, so just 6. And then for all of those carbons, okay? So which is it most likely to be? This one actually has 6 oxygens, so we know that that could be right. So we know that it could be 6 then. Does it make sense that there be a ratio of that many more carbons to oxygens? Actually, yes, it does, because we've got all of these and we've got all of these. Does it make sense that we've got approximately double the amount of hydrogens? Yes, because we've got them all on the top row and on the bottom row of each one. So approximately for every carbon, we have got 2 hydrogens. So that does make sense. And it makes more sense with this amount of oxygens. Okay, so the answer is going to be M, and then you've just kind of got to put that reasoning into some kind of mark. So approximately, we've said approximately, you can draw as a line like that, wavy line. So approximately 2 hydrogens to 1 oxygen in the ratio makes sense, doesn't it? We kind of worked that out. And secondly, that we need, it was only worth 1 mark actually, but also the low number of oxygens made sense. Oh, that is the oxygen, sorry, approximately 2 hydrogen to 1 oxygen. It doesn't make sense, does it? Yeah. It says in the mark scheme approximately 2 hydrogen to 1 oxygen. I just copied it. So what we mean is there's approximately 2 hydrogen to 1 carbon. I beg your pardon. Let's make sure we get this right. So approximately 2 hydrogens to every 1 carbon, because 98 is approximately twice 55. Yeah, not exactly, but just approximately. And then we need the really low number of oxygen. So you could say that there's a really high, to just another way to describe that is you could say that there's a really high ratio of hydrogen to oxygen. That's another way of saying that there's such a low number of oxygen, a high ratio of hydrogen compared to oxygen. Okay, so it's hard to put into words, isn't it? But drawing it out should allow you to work out the right answer. Next one, phospholipid molecules are similar to triglycerides, but they also contain the element phosphorus as part of the phosphate group. Explain how the structure of phospholipids allows them to form a bilayer of plasma membranes. Now, this is tricky at this stage in the course because you haven't studied membranes in any kind of detail yet, but hopefully we know enough about the link between them to answer the question. So phospholipids are often simplified to look like this, where the head of it is the glycerol, like you get in a triglyceride, plus phosphate. And then the 2 tails down here are 2 fatty acids. Now we know triglycerides have 3 fatty acids, that's why they're called triglycerides. Triglycerides were glycerol and 3 fatty acids, if you remember. Whereas phospholipids are just 2 fatty acids, and then the glycerol has a phosphate as well. Now this head here, we often just call the phosphate head. And the tails we call fatty acid tails, just as we've put there. Then you must be able to associate their properties. So the head part of the molecule is what we call hydrophilic, which means that it attracts water molecules. It's attracted to water molecules. Okay. The fatty acid tails, on the other hand, are what we call hydrophobic, and that's because they repel the water molecules. Okay, phobic like phobia. Now, in the membrane, what these phospholipids do is they form a bilayer in order to keep the heads touching water and the tails away from water. And it's not to enable, that's not the right term. What actually happens is if you just were to have lots of phospholipids in water, they assemble themselves in this structure because automatically there's lots of liquid up here in what we call the tissue fluid around the cells, which is mostly water. Inside the cells, we've got the cytoplasm, which is mostly water. So automatically, each phospholipid will spin itself round. So the head is attracted to the water, so that's why it spins, so the head touches the water, and it spins in a way that the tail is away from the water. So the ones near the cytoplasm all line up with the heads on the water and the tails away from the water, and likewise on the tissue fluid, the heads touching the tissue fluid and the tails away from the tissue fluid. Now in the middle of the membrane in here, there's no water, it's effectively dry. Okay, so the tails have repelled away from water, the heads have attracted to water, and they orientate them in this format. And that format is called a phospholipid bilayer, because bi means 2, and you can see that there's 2 layers of phospholipids. Okay, so that's the format that they're automatically going to put themselves into. Next one, an enzyme hydrolyzes a phospholipid molecule to release a fatty acid. What's the name of the bond that's broken in this hydrolysis reaction? Okay, so if we think about a triglyceride, let's just do a recap. Triglyceride, you've got glycerol, 3 fatty acids. So those 3 fatty acids there are going to be joined on in with ester bonds. Lipids are held with ester bonds. Then if we think about our phospholipid, We've still got the glycerol. This time we've got 2 fatty acids, and this time we've also got the phosphate there. So we've got phosphate, glycerol, and then we've got 2 fatty acids. This one we just had glycerol and 3 fatty acids. So this one here is your triglyceride, glycerol and 3 fatty acid. This one here is your phospholipid, glycerol, 2 fatty acids, and a phosphate group attached here. Now, it's just the same bond. Any kind of lipid, these fatty acids are always going to be attached on with ester bonds. So the correct answer there is A, the ester bond. So lipids are always going to be ester bonds. While we're here, let's just do a recap. Glycosidic would be in your carbohydrates joining your sugars together. Peptides are going to be in your proteins. And phosphodiester is going to be in the DNA. '22. Which statement describes triglycerides? So triglyceride, we've got glycerol, and we've got 3 fatty acids. So are they broken down by condensation reactions? No. We know that condensation reactions are always for joining molecules. Hydrolysis are breaking them apart. Are they polymers of glycerol and fatty acids? Now, yes, they are made of glycerol and fatty acid, but they're not a polymer. A polymer would be if you've got glycerol, like for example, glycerol with 3 fatty acids joined to glycerol, 3 fatty acids joined to glycerol, 3 fatty acids joined to glycerol. They're not a polymer. They're not lots of glycerol and fatty acids joined together. They just exist as one. So they're not a polymer. Okay, so it's not condensation reactions for breaking down. They're not polar bonds. Do they contain ester bonds? Yes, they do. Do they contain glycosidic bonds? No, they don't. Therefore, it has to be C. Question number 23. Which of the following formula of fatty acids represents a saturated fatty acid? Okay, so if you've got a saturated fatty acid, it simply means that every single carbon has 4 single bonds with hydrogens on there, and it's saturated with hydrogens. It could not hold any more hydrogens. Now, the other end of your fatty acid is carboxylic acid, so C, double bond OH. Okay, but this is the fatty, this is the bit that gets saturated or unsaturated. So this one here is saturated, okay, because it's all got single bonds. On the other hand, a unsaturated fatty acid, let me draw you one of those. So it doesn't actually matter how many double bonds it has to be unsaturated. You can call them mono-unsaturated if it's got 1, di-unsaturated if it's got 2, etc. Polyunsaturated if it's got more than 1. But the important thing is that you've got at least one of these where you've got a double bond between the carbons. It doesn't matter how many carbons you've got. Now, carbons always have 4 bonds. So this carbon here has got 1, 2, 3, 4. It's already got 4. This carbon here has only got 2. So it's going to have 2 more with hydrogens on. This carbon here has got 2 there and 1 there. So it's got 3. So it only gets 1 more hydrogen. So there's a hydrogen missing in effect just here. This carbon's got 1, 2, 3. Just needs 1 more. Again, there's space for one here, but it can't hold it because it's already got 4 bonds. This carbon, 1, 2, 3, 4. Add the hydrogens. This one, 1, 2, 3, 4. Okay, so this one is what we call unsaturated. Okay, because we've got some space where we could add more hydrogen, but we can't because we've got this double bond here. So what you've got to do is you've got to try and work out the numbers. So the COOH that you've got on the end of each of them is this COOH. Okay, so that's your carboxylic acid, COOH. So that accounts for that part of all of the molecules. Now, the question asked you, was it asking saturated or unsaturated? Let's double check. Saturated. So if it's saturated, for the number of carbons, this one here, for example, has got 4 carbons in that chain there, and it's got 4, 8, 9 hydrogens, so H9. Okay, so the ratio is like times 2, add 1. So on here, 15 times 2 makes 30, add 1 makes 31, so that's the same. Here you've got 17, 17 times 2, well 15 times 2 would be 30, be 34, plus 1 would be 35. So that's wrong, because that should be 35. That's missing 2 hydrogens. And this one again, this one's missing 4 hydrogens. So this is the only one that follows that pattern where it's CnH2n, so 2 times whatever that number is, plus 1. So it has to be just statement 1, and therefore your answer is going to be D. Okay, only one is D. Next question, 24. Oleic acid is a monounsaturated fatty acid found in vegetable oil. Which of the following A to D is the correct structure for oleic acid? So it tells you that it's monounsaturated. That means that we're looking for one double bond. So you've got to know how they draw them in these simplified structures. So the double bonds you should be able to see. Oh, sorry, they've gone on to the next page. Let's just move this down all onto the next page so you can see it all in one go. A bit further down. Okay, so we're looking for one that's got a double bond. So this one here has got no double bonds. Don't count the double bond that's in the COOH. Oops. Okay, don't count that double bond, that's different. This one here has got 2 double bonds. This one here has got 1 double bond. And this one here has got 2 double bonds. You can see each time there's a double bond, it causes a bend in the chain kink. So this is the only one that's got 1 double bond. So C is your answer for that one. Monounsaturated, 1 double bond. Question number 25. Human pancreatic lipase breaks the bonds between fatty acids and glycerol. What's the name given to this reaction? So whenever we break a bond, it's always going to be hydrolysis. Condensation to build the bonds, hydrolysis to break the bonds. So that one should be D. 26. Lipids are a group of fatty or waxy compounds. Triglycerides, phospholipids, and cholesterol are examples of lipid compounds that are important in living organisms. Table 7.1 lists a number of statements that could apply to these compounds. Complete the table by indicating with a tick which of the compounds applies to each statement. You can use more than one tick in each row. Okay, so remember that your triglycerides are your glycerol with your 3 fatty acids. Your phospholipids are your glycerol with your phosphate with 2 fatty acids. And your cholesterol, we don't know much about the structure, but we know that it's a 4-ring structure and there's no glycerol and there's no fatty acids. Okay, it's a different structure. So the first statement was that contains the elements carbon, hydrogen, oxygen. So triglycerides, we know the glycerol is just made of those, that's fine. The fatty acids are just basically hydrocarbons with carboxylic acid, so yes, that's fine. Phospholipid can't be that because we know it's got a phosphate, which is a P. It's definitely not going to be that one. Cholesterol, we don't really know much about the structure, but we have actually seen one in a previous question, so we could look that up. And one of the similarities that we pointed out was that it's still made of carbon, oxygen, hydrogen. Is it soluble in water? Does it dissolve? Does it dissolve in water? So insoluble means it doesn't dissolve in water, and no fats are going to dissolve in water. So that's a tick for all of them. Fats don't dissolve in water. Does it contain glycerol? Nice and easy. These two don't. Oh, sorry, these two do, and this one doesn't. Does it contain ester bonds? This is tricky. We've learnt that these two do. Cholesterol, you probably wouldn't really know, but the ester bonds are what joining the fatty acids on, and that doesn't have fatty acids to join on, and therefore, no, it doesn't have ester bonds in it. Is it involved in membrane structure? Phospholipids and cholesterol are, triglycerides are not. You'll know that much better once you've been through the membranes lesson. And does it contain fatty acids? You can see from the diagram that these two do, and this one doesn't. So some elements of that are quite easy, but some of the cholesterol bits are much more tricky. Number 27, triglycerides consist of glycerol combined with 3 fatty acids. Figure 18 shows a glycerol molecule and a fatty acid molecule. In the space below, draw a monoglyceride molecule. Now, I'm not going to draw the whole thing out. All you have to do is, it makes a molecule of water. So if if I just say I'm going to join it onto that top one there, there's your water, so they disappear. So don't draw those in your final molecule. And then all you're going to do is you're going to draw the glycerol and draw the fatty acid, and you're going to draw a bond, draw them close together so your bond's not massive, but draw a bond from that carbon to that oxygen that's left. Okay, so get rid of what's in my red circle, draw this one up here so it's nice and close, and then a bond from that carbon to that oxygen just there. All right, so every time you just remove the water and draw the bond in. Name the bond that's formed between glycerol and fatty acid. Hopefully we're getting the hang of this. So this is going to be an ester bond. And what else is going to be made? If it's a condensation reaction, it's always going to make a molecule of water every time. Okay, next question. Describe and explain how the structure and properties of different carbohydrate and lipid molecules suit them to deliver their role as energy storage molecules in plants and animals. Okay. So this one here is a 9-mark question. This might be the first 9-mark question that you've come across. Now, it's not going to be a case that you have to actually list 9 things. It's a level response, like the 6-mark questions at GCSE, where they're going to say, have you got a level 1 answer with up to 3 marks, a level 2 answer up to 6 marks, a level 3 answer up to 9 marks. Now, this question, if you read the question, it says to describe and explain, and it asks you to talk about the structure. It asks you to talk about the properties. Okay. And the other command word that we've got here is we've got to describe and we've got to explain. So basically, if you describe the structure, that's one category of answers. If you describe the properties, that's another category. And if you do an explanation, that's another one. So explain is E, S for structure, and P for properties. So if you're looking at the mark scheme, if you've got the mark scheme there, you'll see that in the right-hand column of the mark scheme, it labels the points that you might make S, E, and P, and that's why. So you've got to always break down the question. So you would probably have said, right, I've got to describe, I've got to explain, I've got to talk about structure, and I've got to talk about properties. That's probably how you'd have broken it down. And they've actually just identified it as those 3. Okay. Now, the other thing is it's asked you, obviously, to talk about carbohydrates and lipids. So make sure you do both of those. So if we start off looking at carbohydrates, now the ones that are used for energy storage are the polysaccharides. And polysaccharides are polymers of glucose. So they're lots of glucose molecules joined together. And what you can do is you can hydrolyze the glucose molecules off them. And then you can use those in respiration. Respiration, you can either say releases energy. Releases energy, or you can say makes ATP. Don't say it makes energy. Okay, so it releases energy or it makes ATP. All right, so this polymers of glucose would count as a structure, and then hydrolyzing the glucose molecules off it to use in respiration to get energy, that would that part there is an explanation. So you've said a point to do with the structure and you've explained it. Okay, if we were to look at another property, we could say that polysaccharides are, for example, large molecules. So saying that it's a large molecule is a structural point. Because they're large, they would be insoluble. Insoluble is describing their property. And then you could say, why is it important? Explain why it's important that they're insoluble, is because then they won't affect the water potential of cells. So they won't cause water to move into or out of cells, causing the cells to either burst or shrivel. So they don't affect the water potential. That's a symbol for water potential. So that's then an explanation. Okay, so you can see how if you're making a comment about the structure or the property, you need to explain it each time. Otherwise, they're going to cap what level of mark you can have. You could say another thing about the structure. You could say that they've got 4 glycosidic bonds, and the significance of that is that they're easy to make. So that's your structural point. Then you could say that they're easy to make, to store glucose, and equally they're easy to break. So you could say that's in condensation reactions, and then you could say they're easy to break in hydrolysis reactions to release the glucose for respiration. So there you've explained why it's important that they have these glycosidic bonds. So that's explaining. Okay, you could talk more. I'm not going to write every single thing down, but you could talk about how the carbohydrates are coiled, which makes the coiled shape. They're compact as your structure, and that's important because then they take up less space in the cell. You can store more glucose as a small area, as you'll explain. You could talk about the structure that amylase is unbranched, and amylopectin has a few branches. And you could say that actually in plants, we don't need to release the monomers as quickly as we do in animals. So it's for slower release of glucose, but that's fine in plants. You could talk about how glycogen in animals is more branched for your structure. And then you could explain that by saying in animals, actually, we need a faster release of the glucose monomers. Because we're going to need more energy at once, okay? Then you need to make sure that you do the equivalent for your lipids. So you do a variety for your carbohydrates, and then you go into talking about your lipids. So for the lipids, we're doing the same thing. So structurally, you could say fats have more carbon-to-carbon bonds, or carbon to hydrogen bonds. Fats are really rich in energy. They have more energy per molecule, and there's more energy stored in less space. You could talk about fats being insoluble as your property and explaining that so they don't affect water potential. You could talk about how fatty acids are really long carbon chains. And then the explanation, the reason why that's important is they can be broken down to release 2 carbon acetyl groups, which then go into the Krebs cycle. Krebs cycle you'll come across later in the course. You could talk about how animal fats are often saturated, and they've got a role in protection and insulation as well as energy storage. So what you can do is you can start to build up a picture of how good your answer is based on have you got structures, have you got properties, and have you explained every point. So this, what you need to get in the habit of doing is every time you talk about a structure, name the structure and say why it's important. Okay, that's your explain. Then you might mention a property, and then you need to say why it's important as your explain. Okay, and that's the pattern you need to get into. So what is the question asking you about? Here it's asking you about structures and properties, and it's asking you to explain. So every time you make a point, explain why it's important. Now the way they'll grade it is you'll get up to 3 marks if you basically just said things about the structure. Okay, that's level 1. The level 2, you get up to 6 marks if you mentioned structures and properties, but you didn't explain all of them. Probably you put a few explanations, maybe just for the structural, but you didn't explain the properties, so you haven't explained all your points. And then you'll get up to 9 if you've got a good range of structures and properties and you've explained all of them. Okay, so it's that habit, make a point, explain it. Whenever it says explain in the question, you make a point and then you explain it afterwards. Okay, if you want any more help marking your answer to that question, feel free to ask me to mark them. That's no problem at all. Email them across. Question number 28. Figure 22 shows a triglyceride molecule found in sunflower oil. On figure 22, circle an ester bond. Okay, so they've simplified the drawing. So we know that this structure here is going to be carbon, carbon, carbon. Okay, you can see coming off to the sides of them, the oxygens. Okay, they haven't drawn on all the hydrogens around there. Okay, they don't need to in the simplified drawing. So going from these three oxygens here, which are the three I've just drawn, you can see there's a bond going off to a carbon on another little junction. And then you've got the O double bond O going up, and then it goes off into the zigzag, sorry, that one should be there, fatty acid after that. Okay, so if they'd shown you it drawn like that and asked you to circle the ester bond, hopefully you'd know that the ester bond is basically this line here. Now, when you circle the bond, you don't circle it like that. You have to circle whatever's at either end of the bond. So you would actually circle, in this case here, you would circle round the oxygen because that's at one end of the bond, round the carbon because that's at the other end of the bond, and then because this OH is attached, a double bond O, sorry, is attached, you circle that as well. So that's the bond. So same with glycosidic bonds and peptide bonds. Find the actual line that's the bond. Whatever's either side needs circling, and whatever's directly attached to that as a sticky on the sides, circle that as well. All right, so that's what you should have circled. So on here, find that same bit. So what we're looking for is around here and around there. So you've captured your bond, whatever is either side of it and whatever's just attached directly onto there. Okay, it's quite tricky to identify on the simplified drawing sometimes. Part B, sunflower oil is used to make biodiesel, which contains methyl esters. The fatty acids in the triglyceride molecule are reacted with methanol in a process called transesterification. After the reaction, two liquid products form which naturally separate from each other. The methyl esters float on top of a more dense liquid. Name the part of the molecule seen in figure 22 that forms this more dense liquid. So there's loads and loads of writing in that. All they were actually after out of all of that is glycerol. Okay, let's go through the question again. Sunflower oil is made to make voice. The fatty acids in the triglyceride are reacted with methanol in a process called transesterification. After the reaction, the two liquid products formed naturally separate from each other. The methyl esters, so those methyl esters, look, must have come with the fatty acids reacted with methanol. So that and that have come together to make that. Okay, on top of a more dense liquid. Now, you know that a triglyceride is only made of glycerol and fatty acids. Our fatty acids have gone into this, and it's asking you what is the other thing. So it must just be made of glycerol. That's the only other thing that triglycerides are made of. Okay. Part C, living organisms have many uses for triglycerides, one of which is the production of phospholipids. Name three other functions of triglycerides in living organisms. So you really need to just learn the list of these. So there's lots of things you can say. You can call it a respiratory substrate. That's just a fancy word for saying it can be used in respiration. And that would be obviously to get energy. That's like for energy as an immediate source. They can also be used as an energy store, so energy storage. You could talk about keeping things warm, so you would call that thermal insulation. Other things on the list that they would allow is electrical insulation, so that's around your nerve cells, buoyancy, so helps aquatic animals to float, or the idea of physical protection, so a layer of fat as a kind of protective physical, almost like a padding. Table 22 shows the melting points of some of the methyl esters made from transesterification of sunflower oil and fatty acids. So this is what the start of the question was talking about. Describe and explain the pattern of melting points in these three methyl esters. Okay, so the difference here is that they've all got the same carbon, they've all got the same oxygen, but we've got a pattern here with the hydrogen. So this one at the top has got the most hydrogen, and this one at the bottom has got the least hydrogen. Okay, don't worry about the names. Looking at this, the one with the most hydrogen, look, the melting point is the highest, and the one with the least hydrogen, the melting point is the lowest. So all you needed to do is identify that pattern. So the fewer hydrogens there were, the lower the melting point is going to be. Okay, if we go down a little bit, Okay, you can see they give you a bit of space and it was worth 2 marks. So we're going to need to say more than just that. So let's write that down first of all. So first of all, fewer hydrogens will give you a lower melting point. Okay, now the fewer hydrogens you've got, what does that tell us in terms of fatty acids? What have we learned about fatty acids? Well, basically, all we've found is you get saturated and unsaturated. So if it's saturated, it's got the maximum amount of hydrogens. The less hydrogens you've got, the fewer hydrogens you've got, the less saturated it is. Okay, so we could say that these fewer hydrogen ones are less saturated. That means that they're going to have more kinks in the chain, more bends in the chain, in the fatty acid. Let's to write too close to that box. So there's more kinks in the fatty acid molecule, and therefore they're less uniformly aligned together. So when they try and line the molecules up, the kinks get in the way. So they're less uniformly packed together. Oh, can't write anymore, sorry. Packed together. And therefore you need a lower temperature in order to melt them. Okay, if you have saturated ones, the fatty acids are just like a straight zigzag like that, and they can fit them really close together. As soon as you start to get kinks in them, you've got going up, then it changes direction, might change direction again. You've got another one that goes up, change direction, going up, change direction, You can't pack those closely together. Look how far apart they are. Okay, so the fewer hydrogens you've got, the less saturated they are, the more kinks you get in the fatty acid chains, and the less uniformly they pack together, and therefore the easier they are to break apart. Okay, and that finishes the lipid section.